Hi, okay, histology of the airways and the lungs. Cool, this is a nice one. So what we're thinking of is we're thinking of the passage of air to and from the lungs, probably fairly obviously. So there are a whole bunch of tubes. We talk about the conducting part, and really we're thinking about the nose. What I'm thinking is it's a shame I haven't got any nasal mucosa, nasal epithelium sections. What we'll do is we can look at, I've got 3 slides, we'll look at the trachea, and that'll show us the cells and the tissues and the organisation of that major part of the conducting part of the airway, and that'll be very similar to what the bronchi look like. And then I've got sections of lungs which will have, smaller bronchi, bronchioles, alveoli and that sort of thing. We'll look at the cells inside the lungs and what the lungs look like. And also I've got a special lung section which has a stain for elastin, so we can look at the elastic tissue of the lungs, which is quite important, and we can see how that's arranged. Okay, so the airway, then we have a conducting part and we have a respiratory part. The conducting part takes air into the, basically is taking air to the alveoli, and the alveoli then are the respiratory part where gaseous exchange occurs. And we talk about a respiratory epithelium. That's one thing to bear in mind. So the respiratory epithelium is pretty much the same all the way down the conducting part of the airways, and then it's a little bit different, the respiratory part, because it has a different job to do. The other thing to bear in mind is that the respiratory tract and the lungs form from the gut tube embryologically, which means that the tissues we're going to see, the arrangement of cells and tissues, might look a little bit similar to the gastrointestinal tract, but they have become specialised for breathing air rather than digesting food and absorbing water. But the concept is the same. We have external tubes inside us, right? So we have the external world going inside us and then pushing it back out, in, out, in, out, in, out, which means that the respiratory tree has a job of protecting the body from things that we breathe in and out. Right, we'll see. So, first of all then, we'll look at the trachea. It's a circle. This is a transverse section cutting through the trachea. And like I say, the air to that point has already gone through the nasal cavity, through the pharynx and through the larynx to get down to the trachea. So there are a number of structures and a lot of different things there, but the respiratory epithelium for much of that looks similar to what we'll see in the trachea. But some parts of the epithelium, so that is the cellular lining of that tube, is because there's a crossover with food going past it, some of it's a little bit different, say at the epiglottis and what have you. Anyway, we've still got to do epithelium in detail, haven't we? Let's have a look. So in humans, the trachea has a diameter of 2 to 3 centimetres, is maybe 10, 12 centimetres long, so this clearly isn't human. It's a little bit too small for that. And you can palpate the thyroid cartilage, and then there's a little membrane, and then you can palpate the cricoid cartilage, and then inferior to that, that's where the trachea starts, and then it descends down. If you palpate the sternum, the sternal angle is where the bones meet. That's as far as the trachea runs. So it runs down from the neck into the chest. And because it's carrying air, and because there are pressure changes involved, most of the wall there that we're looking at is a ring of cartilage. Oh, there's the end of the ring of cartilage there. And then posteriorly, so it's a C-shaped ring of cartilage. In this case, almost starts again immediately. So it's a C-shaped ring of cartilage because that holds this tube open, which is vital. Because when you breathe out, you generate negative pressures. You wouldn't want that tube to collapse and slap shut. And also, the cells in here are protected largely by being kept moist by being covered in mucus. So if the tube slapped shut, it would take quite a bit of effort to open up again. So there's a C-shaped ring of cartilage, and the job of the cartilage is to hold all of this, hold this tube open against those negative pressures, those expiratory pressures. And then posteriorly here, we have the trachealis muscle. So running, there's like a flat back to the trachea where the C-shaped ring is joined. That's the trachealis muscle, and that's smooth muscle and elastin fibers. Now, here my eyepieces have a 10 times magnification. I'm looking through the, what's this, the 4 times magnification objective, which gives me a total of 40 times magnification. How much magnification you have, I don't know, because it depends on how big or how small your screen is. But you're seeing a portion of that, so I'm looking at the widest view there. And if we jump up to the next magnification power, we can see a little bit more detail. It's quite pinky. So this is a hematoxylin and eosin stain, so most things are pink and the nuclei are a bit purpley. Oh, that's excellent. That's really good. So up there is the lumen, where the air is, and then up against that is a layer of epithelial cells, supported by a thin basement membrane, a lamina propria, and then we see the submucosa filled with glands, and then we see the cartilage with widely spaced cells, and then we're seeing the connective tissue structures on the outside holding all that together. Let's look at that in a little bit more detail. But as usual, the aim here is to go at a nice relaxed pace, start out wide, zoom in, take our time, very visual, very looky type thing, right? So this is using the 10 times objective, giving me a total of 100 times magnification to my eyes. Let's jump down to the 20 times objective, giving me 200 times magnification. So if we look next to the lumen there, we can see some debris on the inside of the lumen, exactly where we'd hope to collect it. Okay, so the cell layer there closest to the lumen, that's the epithelium, so that's a sheet of cells. Now they look columnar, that is, they look like we've cut sections through cuboids, long, tall cells, and it looks like there's multiple layers there. But actually it's a single layer, so this gets called a pseudo-stratified columnar epithelium, because actually all of those cells are attached to the basement membrane. So underneath all those cells is just a thin membrane, the basement membrane, that's what the cells are attached to. So it's a single layer of cells. Let me zoom in again, we can really see what we're talking about, maybe. It's quite a light stain, this. So it's a single layer of cells, a pseudo-stratified columnar epithelium, and that's what we mean by the respiratory epithelium, and it has cilia. You can just about see that pinkening edge, or I can. Oh, that looks pretty believable. So there are cilia coming from these epithelial cells. So cilia are little appendages from the cell, and they can be moved by the cell. And this gives us the mucociliary escalator. So the other thing we need to look for here are goblet cells. So interspersed oh, there we go. That's a great, look at that. Now you can see the cilia. That's a much better example. So there is our, you can see the cilia there, right? That's our ciliated, pseudostratified, columnar epithelium. That's our respiratory epithelium. And, oh, I can see one. Let's see if I can get it in position for you guys. There's a goblet cell. So goblet cells, they're kind of shaped like a goblet, a wine glass maybe. They are mucus secreting cells. And this is the mucociliary escalator. So what we've got here is we've got a tube inside us. We breathe air in, breathe air out, and of course occasionally you get dust and particles and things that would be bad for these cells. It's a thin layer of cells. Particularly when we get into the lungs, you'll see how delicate the lungs are. So this is a protective mechanism. One of the big roles of the respiratory epithelium is to protect the airway and to protect the lungs. So as you breathe in, if you breathe in dust and particles, hopefully those particles will float to the edge of the trachea, the edge of the tube. They get stuck in the mucus secreted by these goblet cells, and the cilia will lift the mucus with the rubbish inside it up the trachea, back up towards the pharynx, where you can either hawk it out, you can spit it out, or more likely you'll swallow it and get rid of it. This is a major mechanism for keeping the lungs safe and the airway clean. Why can't I feel like I should be able to get this to look a bit better. So that's the respiratory epithelium, and that runs pretty much all the way through the respiratory tract. On this section we can see that some of these cells are a little bit flatter and chunkier, and some places they're a little bit taller. Let's pop back out again. That was it, the 40 times objectives. That was 400 times magnification for me. Gone back out to the 10 times objectives, so 100 times magnification. Now we're looking at the other side of the tube here. You can see the respiratory epithelium at the bottom of the image there. So the respiratory epithelium, like I say, is stuck to the basement membrane, and then the next layer is the lamina propria. And the lamina propria, lamina means a layer, it's a proper layer. There's a load of connective tissue in there which is helping hold the epithelium together, so supporting the epithelium. But then on the other side of that, we're seeing lots of circles, lots of things that jump out to me as looking like glands. They look very glandular, and that's what they are. So this is the submucosa. So the epithelial layer and the lamina propria layer, put those together, you've got a mucosa. The layer deep to that, so the layer sub that, is the submucosa. And in the submucosa here we can see a lot of glands. These are more serous secreting glands, and these are going to secrete fluid onto the surface, onto the lumen of the trachea to protect it. So breathing is very drying, isn't it? You know what it's like if you've had a blocked nose and you've been asleep and you wake up with a sore, dry mouth. That's the air drying out your mouth. That's drying out the cells. This is bad for the cells. This will kill the cells. So in the airway is kept moist. It's kept covered in fluid so the cells don't dry out, so they're protected. And that's a big part of what these glands are doing. That's the submucosa. So mucosa, submucosa, and then up against that, we've now got the cartilage ring. So let me go back up to the 100 times magnification, for my eyes anyway. So epithelium, lamina propria. The submucosa is quite thin there. The glands are a little bit thinner, a little bit smaller, a little bit flatter, but that's what they are. We've also got a few blood vessels in there. And then there's the cartilage. So cartilage looks quite different, looks quite special. There is a lot of extracellular matrix, and there are very few cells. So the cells are the chondrocytes, and the cells are maintaining the cartilage. This is a hyaline cartilage. So they're making the extracellular matrix. They're putting the collagen and the proteoglycans and the gags in there and making this structure. So we've got this kind of slightly flexible, slightly rigid C-shaped tube which keeps the trachea open. On either side you can see there are some cells running kind of longitudinally, the perichondrium, which is supporting the cartilage ring. So that's what the cartilage ring looks like. So that's that layout there of the trachea is what we'll find throughout the bronchial tree. So the trachea ends at the sternal angle by branching into left and right main bronchi, so a left main bronchus and a right main bronchus. Those go into the lungs, and then we have lobar bronchi. So we have 3 branches to the right, because the right lung has 3 lobes, and 2 lobar branches to the left. And then we have more branches to bronchopulmonary segments. We have branching, 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 branching. And every time the airway branches, the tubes get smaller. So if we go outside the ring again, so away from the lumen, the tissue outside the cartilage is the adventitia layer, which isn't staining particularly brilliant on this section. Then we've got to go a few bits in there, but this is the connective tissue and the support tissues. We've got blood vessels and, you know, what have you, nerves and what have you in there. So in the adventitia is around the outside of the trachea, but like I say, it's pretty weakly staining here. All right, that's it for the trachea. To start with, the main bronchi have C-shaped rings of cartilage, and as the tubes get smaller and smaller, eventually the rings go all the way around the cartilage. But we've also got a tube made of smooth muscle, which is an important consideration. Okay, so then we get into the lung. The lung's an interesting thing to look at histologically because it's mostly nothing, but also some good stuff. All right. Oh yeah, that is just very lung. It's not like any other tissue in the body. Looks like a big jigsaw puzzle of space. Now, what we're looking at here is we are looking at smaller tubes, so smaller parts of the conducting part, which are taking air into the lungs, because inside the lungs we've still got all those branches. But the main thing, well, most of what we're looking at here is alveoli. We've got big blood vessels because what's going on inside the lung is gaseous exchange, so we need to have huge vascularity, huge amount of blood, huge surface area of blood, huge capillarization. So we need to have a huge interface, a huge surface area between the air, the gas, and the blood, and that's what we're seeing at the alveoli. They are filled with capillaries, they're super thin walled, and you can that's what we're looking at here. We're looking at lots and lots and lots of surface area. And then occasionally we get tubes like this. We should look at this first because we were talking about the airways and the bronchi. So we've got blood vessels and we've got bronchioles. How could you tell which is which? Well, let's go back to the 100 times magnification. Can we still fit both of them in for me, but not for you? All right, so look at this. This is a wrinkly tube. I maybe haven't picked the best one there, but there's a wrinkly tube, and there's a not so wrinkly tube. Now, I think we can see which is the blood vessel here because the one on the right is filled with blood cells. But we've looked at blood vessels before, and we can see on the right, if I go to a higher power again, so 200 times magnification, and a little bit more light, we can see blood cells in there, but can you also see that this tube is lined by a single layer of thin flat cells? That's the endothelium. And then the cells around that are smooth muscle cells. We've got a little bit of a glancing blow here because you see how we haven't got a circle, and blood vessels tend to make circles. So we've cut through the tube at a bit of an angle, which is why it looks like that. But we've got smooth muscle in the wall there. So there's a blood vessel. That'll be a small artery, an arteriole. Whereas here, this wrinkly guy, well, the reason I say I've picked the wrong one is because that respiratory epithelium, that ciliated, pseudostratified columnar epithelium, as the air conducting tubes get smaller and smaller, as the bronchi get smaller and smaller and smaller, and we get down to bronchioles, that changes to a simple columnar epithelium. So it doesn't look pseudostratified anymore. We've still got those tall cells, and they're probably still ciliated. And then as the tubes get smaller again, we see a simple cuboidal epithelium. The cells become cuboidal. They are as wide as they are high. So if we go to 400 times magnification, if we really jump up here, a bit more light. The epithelium here very much doesn't look like an endothelium. It doesn't look like a blood vessel. All these little inlets and these folds. So this is a bronchiole. And the bronchiole has lost the cartilage. We don't see any cartilage going around here, and we can see that because it's a wiggly, it's a tube that can expand, that has shrunk. That's why it's looking wiggly like this. It looks like it's a shrunken tube, right? We can see smooth muscle cells around it. So that's what those cells to the right of the epithelium are there. So in the bronchioles, we're seeing an epithelium. So the respiratory epithelium has changed a little bit. Those are all sat on a basement membrane. There's a very thin lamina propria holding all that together. And then we have some smooth muscle cells. This is a muscular tube. So because the respiratory tract has grown embryologically from the gut tube, which is a muscular tube, we also find that the respiratory tract is a muscular tube. Also, muscle's quite a good building block for building things, and you can change the diameter of your tubes with this, which is quite useful. And it's this that's a problem in asthma. So this smooth muscle can be triggered to contract by sympathetic innervation, and in asthma something triggers that contraction inappropriately and makes that. Let me zoom back out again. So we can see the blood vessel and the bronchiole next to each other. So some inappropriate trigger causes that smooth muscle to contract, making the airway, all of these little tubes, a much smaller diameter than they should be, making it very hard to push air in and out of the lungs. So that's the problem with having smooth muscle making up the tubes of the airways. But yeah, there's a bronchiole and a blood vessel. Look, well, let's have a look at this one first. See, look, there's another little tiny bronchiole and a blood vessel. Notice how, I can do this time and time again, notice how the bronchiole has a blood vessel with it, so that the airway and the branches of the pulmonary arteries are running together. And you may have come across the concept of bronchopulmonary segments. So a bronchopulmonary segment is a region of the lung that has its own airway and has its own blood supply, and you could take that segment of the lung away and it would not affect the function of any other segment of the lung. And this shows that structure. or that organisation, because we have the airway and the blood vessel running together. They have a matched branching pattern. So if we have a look at this little tiny bronchiole, I think oh, that's pretty, isn't it? So that's at 200 times magnification. That's pretty. I think we could fairly confidently say that here we've got a simple cuboidal epithelium. So the respiratory epithelium has changed. What do you think? So we have a simple cuboidal epithelium and smooth muscle around it. Now the other thing we've got here, so there's collagen of the lamina propria, but the other thing we've got elastin. So we've got lots of elastin fibres in the lungs, and that's what's causing a lot of the shrinkage here. I've got a special stain for elastin. We'll look at that in a moment. But elastin, so elastin helps anchor these bronchioles, these small tubes, to the surrounding tissue of the lung, which helps stop these tubes collapse completely and flatten, which would make it very hard to breathe in. And when you breathe in, it's when you breathe in and out, the tubes get stretched open. And well, you know how when you breathe out, with expiration, it's quite a relaxed activity, whereas when you breathe in, when you inspire, it's a muscular activity. So you're working to draw air in, but expiration feels like a relaxed activity. That's the elastic nature of the lungs. That's the elastin helping push the air out of the lungs, and I'll show you that specifically in a moment. So it's a stretchy molecule, it's a stretchy fibre that's surrounding all of these tubes and making up the walls of the alveoli. And if you have a lung, it's quite squidgy. Yeah. Okay, so now the other thing I wanted to show you was, so if that's our tiny bronchiole, so we're getting to tiny bronchiole there. If we go up a bit, this is a slightly bigger bronchiole, and look, you can still see the ring of cartilage there. So that's a larger bronchiole, but it's not much larger, is it? But that has still got the cartilage ring around it. It's still got those seromucous glands in there. We can see the smooth muscle, we can see the epithelium, we can see the submucosa and the mucosa. So look, there's the cartilage there, surrounded by alveoli. And we've got some little blood vessels. So look, inside the lung, small bronchiole, bit of cartilage, seromucous glands, smooth muscle, and then there's the epithelium. Oh, I can really see that cilia there. That cilia is really pronounced for me. Let me pop up again to higher magnification. That looks great. Oh, we can see the goblet cells as well in there. That respiratory epithelium I was talking about, the trachea, look, see, it's continued along. That structure I was talking about, the trachea, is continued along the airway, so it's a consistent thing. We can see, again, pseudostratified columnar epithelium. We can see lots of goblet cells in there. We can see a cilia border to those cells, very similar to the trachea. So the airway, it's got its structure from the trachea, and then it just keeps branching. Very pretty. And you can really see that smooth muscle there. Look at that. How can the smooth muscle change the diameter of the bronchiole if there's a cartilage ring? Well, the cartilage ring it's incomplete. Look, it's broken up into bits. So that's how the smooth muscle is having an effect. And it's having an effect because the epithelium is wrinkled, right? So the tube is not at full diameter. It's been shrunk. Okay, what about the alveoli then? Well, okay. Well, first of all, let's just jump into any old region of the lung, any old alveoli, and see what we see. We're looking for a number of cell types. We can see very thin walls. So the alveolar cells are called pneumocytes, and we have type 1 pneumocytes and type 2 pneumocytes, and then we have a lot of capillaries in here as well. So if I go up to, this is 400 times magnification for me. Most of the cells that we're looking at are flat, squamous epithelial cells, and those are the type 1 pneumocytes. And we're also seeing some blood cells in there, so there are also tiny capillaries. Red blood cells are passing through there, well, cells of the blood are passing through there, and then we're occasionally seeing bigger cells like that cell up there. So type 2 pneumocytes are larger cells, rounder cells. I think they're spherical, and type 2 pneumocytes are producing a pulmonary surfactant. So you may know about surfactant. Surfactant is a bit like a detergent. It lowers the surface tension of the water. So you need these alveoli to be, these alveolar walls, these alveolar surfaces to be moist, to be wet. The gas that we inhale needs to dissolve into water, so we need to have a wet surface in here. That's also protecting these cells, this very thin surface of cells. But when you expired, when you breathed out, then there was a risk of these very thin walls sticking together, and there'd be quite a big effort to pull them apart again. So pulmonary surfactant reduces the surface tension of the fluids in here, preventing the walls of these alveoli from sticking together, and reducing the effort of breathing. Yeah, so type 1 pneumocytes are the flat ones. We can see some capillaries in there as well. Type 2 pneumocytes are the fat ones. So that's the alveoli. And like I say, most of the lung, most of the tissue is space, is alveoli. An interesting thing about the lungs is if you have a lung and it's inflated, if you cut it, it doesn't pop like a balloon, because you're just opening all of these little alveolar spaces to the outside world. So it's not like a bag of air, it's full of tiny cells of air. So if you cut through to those cells, they'll lose their gas, but the rest of them stay open. Okay, now there's one other cell type I'd quite like to find, and that would be a club cell. But I think we're seeing lots of good representations in here of type 1 pneumocytes, type 2 pneumocytes. We can see capillaries in there and blood cells. These are alveoli, lots and lots of alveoli, lots and lots of chambers running off a corridor. And yeah, lots of blood vessels. Okay. Now, I probably need to find a tiny bronchiole or a terminal bronchiole for this other cell type, the club cell. I don't know if I'm going to... You find most of them... Oh, the other thing you might see, but sometimes you find macrophages inside the alveoli up against the cell wall. So the macrophages are clearing up anything that gets past the mucociliary escalator and gets inhaled all the way down here, which is usually a bad thing. in the cadaver, when we look at a lung, the lung has often got a black speckled pattern, and that's those macrophages. If they've taken up carbon particles and then they get stuck in the lung, that gives us the black marbling pattern that we see in the cadaver. I'm getting distracted again, aren't I? What I'm looking for, the club cells. I don't know if I'm good enough to find any. The club cells, bronchiolar exocrine cells, they're scattered throughout the airway. They're most commonly found in the smallest, the very small bronchioles. They also used to be called Clara cells. They were actually first discovered by somebody else, but that was all forgotten about. Clara was a Nazi, and his research was less than ethical. So you might read about them being called Clara cells in some textbooks. Nowadays they're called club cells, and they're an interesting cell type. So the problem is they're also a cuboidal cell. So they're in this cuboidal epithelium. What I'm looking for is a cell that is not ciliated and is bulging towards the lumen. So these cells are worth mentioning because they have a number of jobs. They are a progenitor cell helping replace lost epithelial cells. They clean up toxins. They manage a lot of toxins and harmful things that get down into the airway. And 400 times again, aren't I? And they produce proteins that interact with the pulmonary surfactant and things like that. So the club cells are an important cell type in the airway, which might be mentioned, but are quite difficult to identify with a normal stain. But can you see how some of these cells are bulging into the lumen, into the space there? Some of these cells look like they have a ciliated border, and some of them look like they have little bulgy bits. Anyway, maybe I'm talking myself into this. Oh, that may be up there. Is that on your... which bit am I looking at there? Maybe. Anyway, club cells, Clara cells. Right, well, that's it, isn't it? We've looked at the respiratory epithelium, the cellular and tissue organisation of the airway and the trachea. We see how that continues into the lung, into the smaller bronchi, as although that airway keeps branching and branching and branching. We've looked at the alveoli, we've looked at the blood vessels. So the other thing I wanted to show you was this elastin stain. So the elastic nature of the lungs. We can look at the elastic nature of the lungs. This is probably a heart's elastin stain? What are we looking at? So there's a light brown staining to most things here. So clearly this is lung. We can see that lung again. And what looks good? Look, there's a larger bronchiole, right? And if we look in there, I jump up, you can see that pseudostratified columnar epithelium. It's not very well stained, it's light brown. We can see a lot of dark speckles underneath it. So the lumen is at the bottom of the screen. Then we've got that wide band of light brown. That's that pseudostratified, ciliated columnar epithelium we were looking at earlier. That's the respiratory epithelium. And then I said there's a thin basement membrane. And then I said underneath that, so deep to that, we have the lamina propria, the proper layer. And I said there was lots of collagen and connective tissue in there. And we're seeing that there's lots of elastin in there as well. So that's the lamina propria. We can see little bits of elastin throughout the other layers, throughout the other tissues, and as we get out there and what have you. But that's just the largest bronchiole. That's not the most interesting bit. If we go and have a look at, oh look, there's a blood vessel. Bump. bump. There's a blood vessel because there's the blood inside it, and we've got a thin endothelium, but this is a quite elastic artery. We've got layers of elastin and layers of smooth muscle there, so we're seeing quite a lot of elastin in the arteries, which we would probably expect. Let's find a little ditty. But there we go. Again, we can see the lumen. We can see the epithelium, and then we can see a lot of elastin as part of the connective tissue. And look down there, those are alveoli. So the elastin are part of the structure of the bronchus. There's a blood vessel. That's all tied in as well. But now if we look at the alveoli, look in there. I can go to a higher power again. And look, everywhere we look, there's a little bit of elastin. In the walls of the alveoli. So there's a lot of, like I say, there's a lot of surface area, a lot of space, a lot of very thin walls. But one thing we're seeing in those thin walls is a little bit of elastin. And that elastin is helping, so that's forming much of the structure. So we don't see loads of connective tissue, we don't see lots of thick layers because gaseous exchange wouldn't work then, but we are seeing quite a bit of elastin, which is part of the structure of the alveolar wall that stops them collapsing, lets them change their shape, and gives us that recoil, that easy exhalation as we pull the air in using our skeletal muscles, and we inflate all these alveoli, we pull air in, and then as we relax the skeletal muscles and relax the diaphragm, all that elastin that you can see there under the microscope was stretched and is now de-stretching, is shortening, and has helped pushing the air out of the lungs. And all of those factors that we've talked about stop these alveolar walls from collapsing and sticking together. So the aim there, again, as with all of these histology videos, is to be very visual and slow. I'm sure it's way too slow for many people. But by doing that, we've seen how the layout of cells and tissues kind of continues with that branching pattern of the bronchioles. It's not super complex. Once you get the pattern, you see that it continues and continues and continues. And the alveoli are probably by necessity quite simple. Anyway, I think it's beautiful stuff. Probably when you just look at this section, it doesn't really mean much because it just looks like mostly space. But anyway, right. I hope that was useful. I found it interesting. I do love looking at this sort of thing. See you next week.